pointing downwards, growing on the undersides of these boulders. Bulbous injured sea star with its arms curled up in the current. Deep sea sole and more of those denier sponges. This little patch of maybe farrier coming up. Got Kurt Whitmire on the science chat as well, who helped plan these dives, commenting on the change in geology and the much larger slabs that we're seeing on this part of the slope. Di is part of the group at the Southwest Fisheries Science Centre that will be doing the analysis of these transects, and she's taking some more detailed notes of what we're seeing as we go along, because that will help the analysts when they come to doing their counts on the video.
Thanks, Abby. I was typing away and I muted so you wouldn't have to hear my typing. Waldo Wakefield has just joined us. Hey, Waldo.
Waldo's also done lots of image-based surveys.
Yeah. So this is working pretty well, lighting-wise and angle. And we've seen some good stuff that we've gotten in this transect.
Dori, do you listen to the audio when you're doing the video analysis? Or is it just the
images?
Yes.
So if anything is called out, it helps the video analyst in case they're not able to see so well. If someone's calling out that's knowledgeable or calling out something that an expert on the chat has identified, that's helpful.
Yeah. The thorny and different type of glass sponge.
I haven't seen any fishing gear, knock on wood.
Yeah, I know there was some concern over an upcoming, potential dive at a site where we saw some aggregating petrale sole. I don't remember seeing any fishing gear on the AUV imagery that we collected at that particular site, but it is always a concern.
Yeah.
That's a feature kind of inshore of the main part of Santa Lucia Bank.
Bob, do you have any concern about this return in Argus on the starboard side? Yep, 20 meters. Okay. And we're crabbing away from it as well. But it has been consistently there.
I'm going to type some more, so I'm muting for the moment.
I think all these little red patches are little tube worms. Retracting when we come close. Big bubblegum coral.
Yeah, we're moving away from that wall in Argus, or ledge.
That's an interesting sponge. Going under the lasers right now. Africalistes, like, possibly.
Another Isabella with a question mark? Yes. And another Swiftia stick coral we'll have to zoom in on after we're done. Hopefully we'll see some more and we can zoom in after the transect.
Yeah. Like to have a zoom on, see that sponge that's just gone past. On the right. We want to have a zoom at.
Shoreside, we're just about halfway through the 200-meter transect.
Thanks. That's Isabella and Heteropolypus. Hagfish down in that crevice that looked like. Could have been something else. Couldn't see it very well. Big slabs of rock.
We're definitely seeing a lot more corals at these depths than we did on the express cruises, where we were up between sort of 200 and 500 meters. From what I remember, we saw quite a lot of sponges, but hardly any corals at all.
Looks like a dead bamboo over to the right. We just passed. You could see the nodes. And these are paragorgids, right? Knobby. Typing some more, so I gotta mute. Unless you want to hear me typing. No!
interesting glass sponges on that rock. Looks like one of them might be on the way out when you can start to see bits of exposed skeleton. It's not a good sign for the sponge. Paragorgia?
We can have Sam do it.
What are you looking for?
The numbers.
Ah. Yes, I am qualified to look at numbers.
Well, and to write them down.
I am also qualified to write them down, yes. Oh, are you serious?
You just told us you were qualified.
Well, Bob, you've got okay. I'm gonna say you've got 70 meters.
Bob, do you need to borrow a pair of glasses?
Oh. Can we?
I think that's really more the concern.
Can we go back on bubble, Bob?
I ain't got cooties.
More heteropolypus. We called those dog toys years ago on our survey. Another Isabella. They're very pretty.
One of those little moon sponges.
It's like there's a lot right up here.
Good
Oh,
spot,
yeah.
some of these little... Lucky we're on Transect. We could have a sampling field day. This is...
All right, should I drop a target to come back to? We are about 60 meters off the end of the transect.
Could you, yes, please, drop a target. There's no harm in that, because this looks like a...
Sure.
Yeah.
What did you call it? A sampling field day?
Yep. We got some thorny head input from Waldo saying at this depth, the larger thorny heads are likely to be short spines.
Yeah.
Which is good because I always find it impossible to tell.
Yeah, unless their dorsal fins are, well, they are separate, they overlap in depth, but the long spine thorny head is smaller. And the third dorsal spine, which you can't always tell it's the third one, but it's taller. That's why it gets the name long spine. But yeah, and one gets to be 40 centimeters, I think 39 centimeters maximum length, and the other is almost twice that, I believe.
Oh, wow. Yeah.
Long spine thorny head is smaller, it's about 40 centimeters max, and it's deeper. And the short spine thorny head is larger, can get up to 80 centimeters in length, or 32 inches, and it is shallower in its depth distribution, but they both, they can overlap in depth here. So the size kind of helps separate them sometimes. When you see thornies that are greater than 40 centimeters, you know they're short spine. Because the long spines don't get that big.
Mm.
Ooh, there's that little yellow
Yeah.
buttons of sponge. Now, did we
Yep.
get some of those in the one sample that we got that had more than one thing in it, Abby?
I don't think so. I think we thought those were eggs in the end, the yellow kind of
Oh, that's
spheres
right.
that were
That's right.
attached. But this chap coming under the lasers too is something that we'd like to sample that looks a little bit like a heterocone. But we're trying
Should
to
we drop a
unravel.
target?
I've been seeing them as we've been going along, so I'm pretty confident
Okay.
we'll come across them all. No, I mean, I think we've hit on an excellent contour. It's worth taking the time to find the right line.
Definitely. Thanks for your help, Steve and Sam.
Mm-hmm. Yep. And guidance from Guy on the science chat as well.
And Guy as well.
And Paul.
Yep.
And Paul.
Takes a team.
Oh, sorry, Paul.
And Bob and Al.
And Bob and Al and the bridge. We don't know their names.
And the bridge is George on the bridge right now. Shout out to George.
Thank you, George. Sorry for all the changes.
I'm sure he's laughing up there. And of course, everyone else
Does
Yeah,
on
the
the
did they
ship.
have a-
Yeah.
Do
Can
they have a
see
video
what,
feed?
we're seeing?
Yeah,
Yeah.
they have a video feed that they're usually playing the Herc cam. And then they also
they
can see
can
high
at
pack.
least they can at least understand why we're asking for.
Oh yeah.
Whoopsie. Yeah. Why we're changing course so often.
Got 20 meters to the end of this transect.
Excellent. This was a good one. An interesting one. And I think the angle works well and the lighting's good and our speed and height are just perfect.
All right. That's what we like to hear.
Thank you.
5-star transect. Would transect again.
Yep.
Is that a plastic bag?
Oh
Looks
no.
like a plastic bag.
Almost did 200 meters without any
Oh
trash.
no.
Can't stop. So we are just about complete. Do we want to stop here and look around, science? We've got about 2 meters left.
Abby,
Yeah, yes, I think so.
I think we should. So you can tell the bridge. Yeah.
Transect is
We'll
stopping right now. Bridge nav. Hold position.
Is there anything that you're seeing you want to zoom in on or sample right now in this?
One of those sponges maybe, Abby?
I think I saw a little clump of barrier, possibly it was just right in the end of the transect, but I think it was maybe to the right where we are now. So this sponge with the crab one is what we sampled just before.
Yeah, I'm working on it. The
Yeah,
current
sorry.
is, Yeah.
Yeah, it looks so ripping.
The current actually was pushing us basically along that transect.
Nice.
I wasn't really
There it
driving
is.
Well,
forward. Oh,
yeah.
okay.
Oh,
Well, that worked well then. Good.
yeah, so we've been sampling Faria from various locations. They're typoglass sponge, they have a fused skeleton, and they're also hard to identify to species level without looking at their spicules under a microscope. So we've been taking samples from the various sites we've been surveying so that we can identify the distributions of these various species.
This current is no
So
joke.
if the current is very strong here, we've been seeing patches of Faria. We have no samples from this depth, so I'm not too fussy about which patch. So if we want to move to find an easier
Well, I think
location to
the current has
zoom and
been pretty
sample.
strong this whole way.
No, just
so
consistently. All right. Okay, well. This is something we'd be interested in sampling, but I'll leave it up to you whether that is a possibility here or whether we should keep moving.
I should be able to set us down. Are we gonna settle here? All right, Al, let's zoom in. And Steve, do you know how we're gonna sample this?
Are we going to sample this shore side? We have confirmation?
Yes, and I would say they're pretty fragile, so I think a slurp has been pretty successful with these.
Okay, well, let's come wide.
Unless you.
Yeah, I mean, unless you think maybe you can get under it and kind of scrape it, and then we can put it in the forward box. We have an open spot. I'll let you decide what you think's easier.
A lot of current. Do you want a laser capture with this?
Yeah, laser capture, please.
A lot of current.
Yeah.
Oops.
Yeah.
There we go.
Can you zoom all the way out and see where we're set up?
You all set, Steve?
All set.
That's a long reach. Let's see. It's pretty far away. I don't think I can get it. Can you look down? I can get it.
All right, jar number 4.
Jar 4.
Oh,
Are you doing the slurp or are you doing
I was going to suggest to grab it whole if you can. It looks so nice.
yeah, never mind.
If you can kind of get under it and
yeah.
cut it and cradle it.
The current's a little bit of a worry, but see.
I mean, it's super fragile.
Hey, can you zoom way in on that mess?
Yeah, I'm gonna need some. There you go.
More? Ship quit moving. Boy, yeah, it's fragile.
You need to scrape that bottom, I think. Go in the forward box on the right-hand side.
It's a lot bigger than it looks.
Lovely.
Ready for
Damn.
Do you need more view of the arm?
Yeah. It's kinda, dang it.
Beautiful.
Nice.
Lovely sample. So with a sample like that, we'll actually be able to see kind of how the tubes connect and look at the structure of the skeleton. Quite often, because they're so delicate, you're just looking at little fragments of the pieces of the tubes.
Steve, what number was that?
That is 043.
Thank you. So
4-3.
science, we have 15 minutes left of the watch. Is there anything here you'd like to take a closer look at as well?
I don't see anything in the immediate area. What about you, Abby, though? There's lots of sponges. That yellow one?
I wouldn't mind having a look at the yellow one just to see if it is the yellow mycale that we see a lot shallower. There are some other sponges that I can't see in this field of view, but I think are nearby. If we could have a quick look at the yellow one. I don't think it's a sample.
Okay. And then we can just keep moving,
Yeah, following that contour.
making progress down the contour line, following the contour. There was some interest in us.
All right, let's
I'm
get a
just
zoom.
chatting while we go in to look. There was some interest in us going down and then coming back up. But we're not going to be doing that on this shift because we've only got 15 minutes left. But that was one thing that was discussed about making our way to 3C. I think we did well on that contour, though.
Thank you for the zoom.
All right, let's
Yeah,
come
I
live.
think that's some yellow mycale. We can keep going.
Okay.
Roger. Bridge nav. 30 meters, bearing 323.
Guy suggests that we stay on this contour as well. Thanks, Guy, for that confirmation. Appreciate it.
Keep a lookout for those glass sponges. They look like heterocone. They're white. They've got these finger-like projections all on the outside. They're quite close together, and the inside of the sponges looks like, yeah, someone's just poked their finger all over the inside, and they're kind of growing in a funnel shape.
Okay.
Is that one of the sponges that you saw in the sponge field day?
Mm-hmm.
Okay.
Yep. But we've been seeing a few of them kind of along here.
On the hunt.
Yep.
So that ship moves in. Is that a big chunk of dead sponge with other things on it?
2 other living sponges on it. Yeah.
Oh, yeah.
Yeah.
And coral, right? Coral as well.
So a lot of these glass sponges, when they die, they'll just fall into a little mat of spicules on the seabed. So there'll be kind of no trace that they were ever there. But for some of the sponges that have a fused skeleton, the skeleton sticks around and other sponges will grow and attach to it. It's a small Garcinia, pink fluffy coral on the rock.
Oh, yeah. And another, we passed over another bamboo. Have we seen many bamboo corals on our dive so far down here, Abby? I don't recall.
Those aren't
I
the ones
remember
at
seeing
the
a
top
lot
of
of
the screen.
Oh, yes,
or
yes,
Steve
it is.
remembers. Yeah, oh, good eye. There were lots of bamboos on the Davidson dives, of course, that were so deep.
Is that a
So-
heterocone sponge? Top right?
Yeah, so there's two of them.
Two.
There's another one off
Can you
to the
see
left.
from here if you have a
Yeah.
preference between the 2 of them?
Do you know, I think maybe the one on the left, just because it looks like it's got some damage to it. So why not take a sample of that one? It's hard to tell from this distance, but the fact that I can see some brown patches looks like it's maybe got some dead tissue. But if we could sample some of the not degraded tissue on the damaged sponge, I think that would be the way to go.
Ship's holding position.
Copy that.
Excuse us, crab.
Yep. Yeah, so if you look at the heterocone calyx that we got some samples of up near Grays Canyon off Washington, the morphology is very different to what we're seeing here. So this might actually be a completely different species and possibly different genus.
Where would you set down here, Bob?
Right in front of the crab, I guess is good. This is a grab again, or is this what are you doing with this?
No, this is the sponge at the top of the screen.
Yeah, I'm not.
Is this a grab? Not a crab.
That is a crab.
I hope he recognizes a crab.
We're not gonna grab it.
Excuse me, crab. Can you grab a sponge, please? Yes, a grab.
One of his pincers is broken off. Looks like. I don't think it's supposed to be shorter.
I always have to have the basket that don't run.
And we have 9 minutes left of watch.
This could also be, this might end up being a slurp. I think they're quite fragile.
Hmm. Do you think it's a grab and slurp or a?
I know that the heterocone for the north, when you take them, it's almost like trying to sample bits of tissue paper. So the grab the bits just float away as you try and move them through the water column. This one might have a slightly different texture because, like I said, I think it might actually be something different. So it's a bit of a gamble. I think you can definitely slurp some pieces. I'm not sure whether a grab will give you something substantial to hang on to. Getting some good zooms would be ideal. Oh, there goes the crab.
Crab wanted none of that.
Mm.
Nope.
This current is making it not easy.
Well, so you wanna push forward into the rock and down at the same time, and then do the stick lock. There you go.
Are we steady there? All right, let's zoom in now. All right, let's come back wide.
Lasers.
Yeah.
Get 'em.
Beautiful.
Got it.
Yeah.
So this is a
It's a
slurp.
little predatory sponge up to the left of it. Mm.
You want to slurp this? Short side?
Abby, what do you think?
Yeah, I think a slurp.
Char 4?
Yeah. Oh.
Light up on there. That's annoying.
Yeah, the indexes are kind of.
Oh, 3. Which way is 4?
The other way.
Wow. All right.
All right.
Yeah, that's it. That's the one.
All right.
So Paul's giving this one a whirl.
How do you move the seat back?
You gotta pull the yeah, there you go. Doesn't move back very far though.
Yeah.
I usually get the arm out of the way because it's
Oh, okay.
like right in your way. You gotta sit a little bit side saddle, I think. I don't know.
Can you help?
Sure which one it is. This feels really loose.
You're
Where's
trying
the-
to swing the arm up?
Yeah.
It's the button.
Oh, there's the button.
You gotta push the button in. It's right at the joint right there. It's right, that thing right there.
There we go. The first test.
All right.
Want me to do this?
Yeah. Can we zoom in a little bit, Al?
That's all the way right. No crashes.
Can we come wide a little bit, Al? Yeah, let's zoom in. Thanks. I think. Are we ready for section? Yeah.
If you could try and get the avoid the patches that are brown.
Oh, yeah.
Yeah.
Yeah, is that? All right.
I don't see the slope chart, but if those went in, that's probably
Yeah,
great.
they're in.
Brilliant.
Yeah. Let's come wide out.
I am going to exit the V-Link. Thank you all so much, so I can let Lisa and Meredith on. Good night and thank you.
Good night, Di. Thank you.
I'm watching those little sponge pieces fall into the jar. Thank you very much. Great sample. I'll sign off as well so the next watch can sign in. Cheers, everyone.
Good night, Abby. Thank you.
Thank you.
Lisa. Hi, good morning. This is Lisa, or good evening. Can you hear me?
I can hear you, Lisa. This is Meredith.
Hey, Meredith, how you doing?
I'm good.
Guess they just collected a sample of the header cone, so that's great.
Yep, Abby's been keeping her eye out for these on the last transect and happened to end the transect basically right at a bunch of them.
Great. It seems from
Video, if
looking at
you're
the
there,
side
can you
chat-
zoom in on the carnage?
Yes. Let's zoom in on the carnage.
They're on a handover.
Okay.
Zoom in.
Is that you, Dan?
Yeah, that's me. I'm messing with everybody this morning. Heckling Paul for that sample, which was beautifully done. And now heckling the video guys because they're trying to hand over. What are we doing?
We'll just wait for them to make
Who
it
am I
happen.
talking to this morning? Is this the regular crew here?
Yes, it's regular. It's Lisa and Meredith.
Lisa and Meredith.
Good morning.
Yep.
Hello.
Good morning.
We'll do our introductions when everybody's on.
Yeah, sounds good.
Should we run that jar around to, let's run it for another minute and then, yeah, I can do that. Which one of these buttons do I push? 6, right?
6. Yeah.
Just gonna run that suction sampler for a minute and make sure everything's in the jar, and then we'll move it around to the flush so it doesn't escape.
Hey guys.
Hello, Steve.
Hey, good morning.
Are we zooming here?
Oh yeah, I was gonna look at the sponge while we were waiting close up, but. So Paul will be down in the galley now so he can appreciate his fine sampling abilities. Let's see, sample jar reverse. Oh, that's fast. What are we doing this morning?
Well, is everybody on?
I think so.
Yeah,
Steve's
so why
here,
don't we do
Jake's here,
introductions
Rennie's here,
Yep.
Dan's here,
Yeah.
Taylor Ann's back there, I think.
Hey, okay. So Taylor Ann,
Somebody's back
Steve,
there, I can't see.
Rennie,
Oh, she's back there.
Jake, and Dan, right?
Yeah.
Yep.
Okay.
The Fab
And
Five.
So my name's Lisa Loney. I work for the Office of National Marine Sanctuaries at the West Coast Regional Office, and Lisa is.
This is Meredith Everett. I'm at the Northwest Fisheries Science Center, where I'm a geneticist working on corals and sponges.
Okay, so the plan for
Sorry, you're breaking up quite a bit.
Yeah, we're not getting any of that.
Okay.
Sorry.
Do you want to?
Oh, better now.
Should I sing a song and we can fix it that way? What are the things that I need to do?
Wish I could tell you.
So let's try it again. Go for it again.
Okay. Sing a song. Sing it loud.
As
That's
soon as
pretty
you
good.
start talking about
That's
it, it breaks up.
okay. I don't know what to do. I did restart my computer. Should I restart again?
Well, for now, how about I pass along what I have, and you can tell me if that is what you'd like to do. So we're between 3B and 3C right now, and we're following this contour near the top of the slope, following it along towards 3C, kind of taking it slow and taking a look at what we've got along the slope here, yeah, so that's kind of straight for a while, about 400 meters-ish. We'll be going roughly the same direction.
Yes, that sounds good. And occasionally we should peek up over the slope again to see what's going on there, and then we can go back down to this contour. Does that sound okay? And we'll make our way to 3C.
Sure, sounds good. Just let us know when you want to make a detour upslope, and we'll just start heading along contour. If there's anything else you need right here while we're stopped, now might be a good time to take care of that while we're not moving along. But we are only at 800 meters depth, which is not so bad as far as ship movements go. Not as bad as 3,000 meters. We'll be able to stop and start at will. So.
Excellent.
Okay. And Meredith, you should let us know about any corals that you would like, coral specimens that you would like to collect while we're on this track to 3C.
Yes. Okay. I think I'm not seeing anything right in the immediate area because they already sampled one of these single bamboos earlier. So we could probably just start moving, and I will try to keep an eye ahead to if there's anything I think from our sample list.
Okay.
All right, so we'll be crabbing along. I'm going to send the ship 323.
323, you're under.
They were kind of crabbing along. Bridge
Roger that. Do I still sound not so great over V-Link?
It's decipherable now, so not as bad, but it's still a little choppy.
I'm going to log out and log back in then.
Okay, thanks.
Okay.
Wow, some current.
Yeah, that was mentioned a little bit. I don't know if Bob passed that along.
Yeah, it's been ripping for a little while.
Sweet. Ship moving yet, or?
Just called it in. Hasn't started yet.
I was not paying attention.
Okay.
Okay, this is Lisa. I'm back. How does it sound now?
Sounds okay.
See that coral there waving in the breeze.
Coral strumming.
Yeah.
You know when the current is high when those paragorgia are vibrating. They're pretty stiff.
Yeah.
That's got all its polyps in, not bothering to attempt to feed at all.
There's a thorny head. Can't tell if it's a long spine or a short spine.
This is a tricky depth because they overlap here, so it could be either.
Oh, strange.
We've got a petal sea cucumber just to the left of the coral with a white frill. That's a species of sea cucumber that's what we call sessile. It attaches to the rock and it doesn't move.
I chimed in when they first got to this fault scarp, and they've seen those petal sea cucumbers the entire time. There are a lot of them along this transect. So if they are sessile, they don't have the tube feet to move along?
Nope, I don't think they have the tube feet to move along. They just have those front feeding appendages.
Right. So they are sea stars, and I think most people know that sea stars have 2 feet, and so most sea cucumbers also have 2 feet to move along the bottom. But because this species is a sessile, that means it's attached to the sea floor. It does not need them because it doesn't need to move. It waits for the food to come to it using those frilly appendages or tentacles.
Yep. And sea urchins, which we may see some of tonight, also have 2 feet down in amongst their spines if you get a really close up look at them.
I haven't seen any sea urchins, but maybe we will see them.
Got a deep sea sole there on the left.
First fish of the watch, I think, unless somebody saw one earlier.
We've had a couple thorny heads.
Oh, that's right.
Yes.
Blocked them out.
Yeah, they're just, regular feature. So that was the deep sea sole. Is that what you said, Meredith?
Yep, that's the deep sea sole.
Mushroom coral, a couple of mushroom coral.
Yep. Our Heteropolypus ritteri. Occasionally you'll hear one of us call those Anthomastus because until about 2 years ago that was the genus, but they've been moved to a new genus.
Do you know, based on what they were moved to a new genus, what was the distinguishing characteristic?
I think just a little more detailed taxonomy was done. Anthomastus is a species that I think is more in the Atlantic. I think that a little more taxonomic work was done and a little bit of genetic work, and it was found that they're just not that close to the Atlantic species, so they moved them to their own genus. Looking at these deep sea corals and sponges, a lot of them in the Pacific and actually in the Atlantic, a lot of them originally got their names based on comparisons to shallow waters taxa. And then in the Pacific, because research out here is newer than research in the Pacific, a lot of the same thing happens where a lot of the groups out here just get named to things that they match in the Atlantic. Although then as we do genetic work and do more detailed taxonomy with more modern resources, a lot of these are being moved to their own groups because they're not actually, in fact, that closely related to the groups in the Atlantic.
There's a skate or a shark egg casing.
Oh yeah, right there. What is the I've seen what is Gersinia? Which